One about my research in Antarctica, and the second one work closer to my home in Scotland. So I hope you don't mind, it will be quite informal. Please ask me any questions as we go, that's no problem. And I want to tell you a little bit about the experience of working in the polar regions as well. So please do shout up if you have any questions. And if I speak too fast, please tell me to slow down, it's no problem. Okay, so I will be talking to you about Antarctic bryozoans and how you work on these species underwater. So what I'll be talking to you about first of all is I will introduce you to Antarctica, why we're interested in Antarctica at the moment and what changes are happening in our oceans, and why bryozoans are interesting to tell us about these changes. The project itself, which I was going down to Antarctica to work on, and the practicalities of working in this kind of an environment. I'll tell you a little bit about the results from Antarctica, and then also a surprise data set from the UK, and a comparison between the UK and Antarctica as well. And then I'll talk to you a little bit about the summary, and then the next steps of this research. So first of all, Antarctica, I'm sure you all know where it is. It's the one in the south. It's the highest, driest and coldest place on the Earth. And it's 1.5 times the size of the USA, so it's very big. And it is mostly covered in ice, with an average of 1.8 kilometers thickness, so a lot of ice and snow. It's very, very dry with a very low average annual rainfall, and that includes snow. And there's significantly less precipitation, rainfall and snow than there's inland. It's been evolutionarily isolated for 25 million years, and that's because of the circumpolar current which runs all the way around here and stops anything escaping. So it's got some really interesting creatures living there because of this. It's extremely seasonal, just like here. It doesn't get dark at all in the summer, and it doesn't get light at all in the winter. And it has temperatures down to minus 89 degrees centigrade in the centre. Has no permanent residence, although some countries are trying to have the first babies born on Antarctica so that they can claim the land. Chile has been flying pregnant mothers to parts of Antarctica to try to claim the first residence. It's very interesting. And because of the Antarctic Treaty in 1959, Antarctica is untouched. It is not used for oil, and is not used for mining minerals. And there can be no armies or navies held in Antarctica. So at the moment this really protects Antarctica as a really interesting place to research natural environments. So as you probably all know, there are many changes that are happening in our oceans at the moment. Temperatures are getting warmer, and they are also becoming more acidic, caused by carbon dioxide in the air absorbing into our seas. And this is happening faster around the poles than anywhere else. So there is more warming, and it is becoming acidic faster. And this is causing a lot of loss of sea ice. This is a picture that NASA took from one of their satellites. And you can see this shows the increase in winter temperature over the last 25 years, and you can see how much the peninsula in particular of Antarctica has got a lot hotter over the last few years. So it's an interesting area to look at the moment, and this is the fastest warming place on the planet right now. So why bryozoans? There are a lot of bryozoans in Antarctica, they are everywhere. They're also very what are bryozoans? I shall tell you first of all. They are small filter-feeding animals that live together in a colony. So they build a shared calcium carbonate skeleton which they live together inside. And they come in many different shapes and sizes. Their skeletons vary a lot. This is just a few examples of the different types of skeletons that these have. But they all have these small units within which one animal will live, and they will live together in these colonies. And they're interesting to me because their skeletons are made out of calcium carbonate mostly, and of two different types of calcium carbonate. calcite and aragonite. These are chemically exactly the same, but the way that the molecules are held together mean that the crystals are very different shapes and they have different properties. They also incorporate other minerals inside calcite in particular and aragonite. In particular, they hold magnesium inside calcite, which I will be talking about later. And this is some examples of the different types of crystals that the skeletons are made up of. So there is a lot of variation, and this is all dependent on the chemical composition of the calcium carbonate itself. So we have here, this is mostly calcite, no aragonite at all. And here we have more aragonite needles, which is smaller and thicker. And why does magnesium incorporation vary in bryozoan calcite? Well, there are a few different theories which are around. First, people believe that it is species-specific pre-programming. It is just something which is genetically inherited within a species, and it doesn't vary beyond that. Other people think that it is varying down to biological factors, such as when the animal gets older, it will have more magnesium. Or the growth rates, how fast it is growing in the colony, and how much it is able to feed. The most common reason given for the variation of magnesium is changes in environmental conditions. So it has been shown that in some marine species, when the temperature increases, magnesium increases in the skeletons. And because of this, it is very popular to use this to try and understand paleoclimate. So looking at bryozoans in fossils or other species in fossils, and seeing what the temperature would have been like at the time that animal was alive. And it's also potentially useful to look at long-term environmental change, such as ocean warming, because we can see if the skeletons are changing chemically between now and maybe 100 years ago, and see whether this is causing any effect. So I wanted to assess whether this could be used for monitoring the effects of climate change in Antarctica, whether this magnesium varies in a way that we can find useful. So this study is all about looking at whether it's down to genetics or whether it's down to environmental conditions. So the project aims to look at the skeletal composition of common Antarctic bryozoan species, and also some from the UK, from temperate waters. And I wanted to look at the effect of local environmental conditions on skeletal chemistry, and also find out how much variation is specific to each species, and compare this for Antarctic and UK bryozoans to see whether the species vary different amounts naturally in the environment. So to do this, I collected many specimens of each species from the bottom of the ocean by scuba diving. They were very fragile, and they all grow encrusting on rocks. So it is very difficult to collect them by a net or by dredging without damaging the fragile specimens. So we did this in both Antarctica and also in the UK, and also collected environmental measurements at the same time. We collected these in a very short period of time to try and minimize any effect of season, whether it's summer or winter or anything like this. So this is what it kind of looks like. I don't think you can see this very well, but this is me underwater dragging lots of rocks along the bottom. It's not very elegant. I was also collecting water samples, and I don't know if you can see this, but I also have loggers, electronic equipment attached to me, which recorded the temperature.
Yeah.
Is that a bit better? Okay. So yes, I have loggers here which are attached to me, which monitor the temperature and also the salinity during the whole time of the dive. Once I had the skeletons, I examined it using a technique called X-ray diffraction, which looks at the chemical composition and also the array of the molecules within the skeleton to see how much magnesium was there. And I also took very fine microscopic pictures of the individual crystals themselves within the skeletons. So where did I go in Antarctica to do this work? I went with the British Antarctic Survey, who have a few different bases, and I went to Rothera, which is their main all-year-round base. So this is occupied in both winter and the summer. It's been there since 1975, and it is the biggest base with about 22 in the winter and 100 staff during the summer. So it's the biggest one. And the most important thing for me is that they maintain a year-round dive team, so that you can always dive there, in theory. It's on Adelaide Island, on the peninsula, and it is heavily glaciated. So you will see this later, that there is lots of ice in the sea, which comes from the glaciers, mostly. And this is what the base itself looks like. So it is it has a runway, which is very important. Not many of the Antarctic bases have their own runways. And it has a number of different buildings. Down here is the scientific laboratories. This is where the planes are kept. And then these are the areas for living. So it is very close to the sea from all angles. And it has a number of small bays here, which are very good for sampling. So whatever the weather, usually you can go in one of these bays. So I just wanted to show you a small video of what it's like when I arrived. I don't think you'll hear any music, but that's okay. So we fly to Antarctica in small planes with propellers, and we go from the very south of Chile, and it takes, I think, 3 or 4 hours. I was there during the height of the summer, so it was the warmest time of year, and also the lightest time of year. So this is the closest I saw to sunset the whole time that I was in Antarctica, and I'm actually landing at 2 o'clock in the morning. Okay. So, a bit about the practicalities of diving, the more fun stuff. So what's it like diving there? First of all, it's very cold. This is not surprising, I don't think. The warmest is about 0 degrees, and it gets down to minus 1.5. So yes, you have to be prepared for that. The second thing to consider is that there are predators. There are things that will try to eat you. Orcas, or killer whales, are one problem, but the biggest problem we have at Rothera are leopard seals. These are like normal seals, but huge. They are about 3 to 4 meters long, and they weigh about 600 kilos. And they are very aggressive. We actually, at this base that I went to, the last person who went to study bryozoans, the last female, was actually killed by one of these when she was diving. So it is something that they are very worried about, and we have to take many precautions about these leopard seals. So because of this, before every dive, somebody has to go and sit in the cold and watch for half an hour to make sure that there are no leopard seals or whales in the area. They also stay there the whole time we are diving to make sure that it is safe. And if they see any whales, then you cannot dive for that day, or leopard seals. The next thing to be worried about are icebergs. And they are very beautiful, but they are also really big and really heavy. And they fall over a lot. They are very unstable. So when you're in a very small boat like this, which is what we use for diving, if you are too close to an iceberg and it falls over, then it will capsize your boat. The other thing to worry about with icebergs is that they scrape the bottom of the sea as they move along, and they move very quickly. And this is called scour, when they scrape everything clean off the rocks. And that also means people. So if you're in the way, you will be squashed very quickly. Yes. Because of the icebergs, we also have to put our boat in and out of the water between every single dive, because otherwise your boat gets squashed by icebergs coming into the marina. So it takes a lot of time. And because of the cold and the predators, we had to have some special equipment. So this is what we had to wear to go diving in Antarctica. This is not me. The first and most important thing is a full face mask. This covers all of our face, but it also means that we can talk to the other scientists underwater and also talk to the people on the boat. So if they see something, then they will tell us to come back quickly or to get out of the way. So that's very important. The next thing that's important is these ropes. So you can see one here, which is kind of coming towards you, and there's another one back here. Every diver is roped to their buddy, their other diver, and also to the boat. So it's very easy to make diver spaghetti because you have a lot of rope in the water, and it's very easy to get tangled in all of this. Of course, we have to wear lots of warm clothing, so we use dry suits where we only have air inside, and they are sealed at the wrists and also at the feet, and so we can wear our normal clothes and thermal clothes underneath and stay warmer. And then, of course, the most important thing is the stick. So we do not get given any weapons at the British Antarctic Survey. We are given a stick. I'm not sure this would be very useful if you are attacked by a seal or a whale, but they like to think it will help. But because we always have to take this, and again it is very annoying and awkward and big, we use this for science. So we mark it with distances, and we use this to measure things. So it's not so useless. And I'll just show you a little video of, maybe, I think, yeah, of what it's like when we actually go out for a dive. So every time we have to put the boat in and take the boat out. And it's a very small boat, so we can only have 2 divers at any time. It's very difficult to put on any of the equipment yourself because you have thick gloves, so you always have people to help you put things on as well. Sometimes the ice is really thick when we are out in the boat, so we have to go very carefully in case the boat hits an iceberg and falls over. We are actually collecting icicles here for our drinks, I'm afraid to say, but yes, it's an iceberg. And of course, sometimes we get to see nice penguins that come to us. So these are a few pictures of what it's like underwater, because although it's very cold and the icebergs are scouring all the time, there is actually a lot of life underneath the ice. There's, oh yes, that's just a diver, lots of ice. So you can see the difference where the icebergs have passed quite recently. So here is an area where an iceberg has been quite recently, so you can see there is not so much life. And these areas which are more protected from the icebergs and have a lot of sponges and a lot of soft corals. There's also a lot of different weeds, and you can see that the water is very clear, so they grow very well, even at quite deep depths. And a lot of this is bryozoans, so I was very happy.
And how deep do you go and how dive?
Only 25 meters. So we were limited to this depth by safety. So we could go deeper, but we don't want to hurt ourselves. So although we have a doctor and we have a special chamber for treating divers if we got sick, we don't really want to have to use this when you're in the middle of nowhere. It also gets colder the further down you go, so it's not so much fun. Most of my work was done at about 10 or 15 meters, although I did do some of it deeper. So yes, you can see there are many bryozoans. Every time you turn a rock over, it often looked like this, with all kinds of different things. And all of these are bryozoans, and all of this is bryozoans, and all the white patches back here. So it is heaven for me. So, a bit more about the place that I actually went to. As I said, it was on the peninsula of Antarctica, and here is the research station. You can see that's where the runway is. So there are a number of islands around the area, and I took sites at each of these, and took lots of rock samples to compare between these different sites. And I was working on 3 different species: Antarcticothoa antarctica, Hippodonella inoma, and Fenestrelina rugula. All of these are quite common species in Antarctica, so I would not be harming the environment by collecting lots of these, and also they were quite easy to find in each of the sites. So the results. First of all, I also did a little bit on a 4th species, Inversiula nutrix, but I only did a small amount, so I haven't mentioned it here. You can see that the skeleton chemistry is different between the different species. So although these two are quite similar, the other ones are quite distinct. But there's quite a lot of variation between the different samples, even though these were all colonies of bryozoans that were the same age, collected at the same time. There's quite a lot of variation in mineralogy. And if we look between the sites, there was quite a lot of variation between sites. And here I looked at both sites across a whole 10-kilometer scale, and then I also looked at 3 sites just 10 meters apart from each other, so very close together. But there's still quite a lot of variation. And I've extracted just the results for Fenestrelina rugula, so you can see it a bit more clearly. So even though these 2 sites are only 6 meters away from each other, you can see that they're quite different skeleton chemistry, which is interesting. And statistically, all of the species had different mineralogy at all of the different sites, which was an interesting result that I wasn't really expecting. And no relationships were found between the skeleton chemistry and the site depth, or the sea temperature, or the salinity at the time, which was a bit of a surprise, because we thought that it would be explained by temperature, like has been seen in other types of animals, and what is a relationship which is relied on for paleochromatography. So if anyone's interested, this research is published in this paper, so you can always go and have a look. So, is this just an Antarctic thing? We wanted to try this experiment somewhere else as well, to see whether or not this was just a specific type of variability that we only saw in the polar regions. So I replicated this experiment in Scapa Flow in the UK. So this is the UK, and right up here is Scotland, where I live, and right at the very top are the Orkney Islands, and this is where Scapa Flow is, a bay right in the centre of these islands, which is about the same size as the bay that we looked at in Antarctica. Obviously it's very different in all other ways, but it's the same kind of size and the same kind of depth. And again, I took 5 sites, which were approximately the same distance apart from each other within a 10-kilometer area. And I chose 3 species, as closely related as I could find to the Antarctic species, and looked at their skeleton exactly the same way. And we found pretty much the same thing. First of all, the 3 species were all different, and they were all quite variable. even though, again, the specimens were the same age from the same place. And these are just previous publication analyses, so you can see that they're in the same kind of scale as my ones. And when we looked at the different sites, again, we saw that there was quite a lot of variability between sites for all of the different species, looking at it for just the one here. And the same thing happened again. For all of the different species, they're statistically different from each other at the different sites within their place. And again, there was no statistically significant relationship with temperature, salinity, or any other seawater chemistry factors. And if you want to have a look at this stuff, this is all published in this paper. So I was quite interested to see whether the variability which we saw was different between the Antarctica and temperate regions, in this case the UK. And so I wanted to just look at the scale of skeletal variation each species encountered. Now, just to look at the different climates, if you like, this is Antarctica on your left, and this is the UK on the right. So as you can see, this is much colder and much more stable. So the annual variability in temperature in Antarctica is just a few degrees. It never really varies very much in the seawater. By comparison, it varies much more in the seawater in the UK. So these are other factors, looking at daylight here, and looking at things like nutrients, and here is ice cover or precipitation. So based on the paleoclimatography scale, I expected that you would see more magnesium variation in an area where you see more temperature variation, because magnesium is meant to be related to temperature. So this was the theory that I had. However, when we looked at them, they're exactly the same. There is no statistical difference at all between the amount of variation in a species skeleton in the UK and in Antarctica. So, to summarize, there's a high level of skeletal variability between sites, especially in Antarctica, and there's much more variability than we expected. And we were wondering if this might have any implications for climate change. The differences in magnesium calcite between the sites were not attributable to temperature, salinity, carbonate chemistry, or depth, or really the distance between the sites either. And this raises some implications about using this kind of measure for paleoclimatography. If we are wanting to look at skeleton chemistry to tell us what it says about temperature from 200, 500, 5,000 years ago, it's not very helpful if actually it's always changing at the moment anyway. And the other surprise was that Antarctica and the UK are remarkably similar in skeletal variability within the species, despite the differences in the seasonal environmental stability in the regions. We would have expected there to be much less variation in Antarctica compared to the UK, given the temperature difference over the year. But we didn't see this at all, which looks like there was something else controlling this variability. So I've set off a lot of further experiments. So in the last couple of years, I've been doing 6 more species, 8 more sites within Scotland, and we've been looking at the way that the skeleton variability changes over a 3-year period to see it through the seasons, and looking at a much higher resolution of environmental factors. So we have equipment in the sea measuring every 10 minutes for 3 years. So the experiments are finished now, and they will hopefully be getting published quite soon. But as a sneak preview, we still see the same variability. And my conclusion is that bryozoans are no good for paleoclimatography. So, okay, thank you very much. That's the Antarctic talk finished. So I have to thank all the people who funded me. I had quite a lot of funding from different places because Antarctica is expensive. So, yeah. Does anyone have any questions for me? Yes.
Quite disappointing, isn't it? But you spend
there's
a lot
no
of time doing. No, probably chemical composition is not only a way to study paleoclimatology. You know about the
oh yes
the size and different
yes.
other stuff. So particularly this method or particularly this idea was not productive. Obviously.
No, this method
But
is
what about the relationship between the calcitic and aragonitic skeletons, for instance? This could be
no, not really.
Not really.
I didn't show it here, but for the temperate species in the UK, they are actually both calcitic and aragonitic, some of these. So I was able to look at the ratio of these two to each other. And in my next talk, I will also talk a little bit about a species which has both.
Mm-hmm.
And aragonite is a bit of a better measure for temperature, but it's still not completely consistent. So I don't think it's very reliable. I think it is more to do with the metabolics of the animal and how happy it is. You know, if it is under stress and it's trying to save all of its energy for surviving and for reproducing, then it doesn't put the energy into making the more expensive parts of its skeleton, which are aragonite and magnesium calcite. So I think the rule that magnesium follows temperature is probably a bit simplistic because it is based on the underlying metabolism and the assumption that all animals are happier when it is warmer. But of course not all animals are happier when it's warmer. Some animals are happier when it's colder, especially if you live in Antarctica, you don't like getting warm. So yeah, I think that there's some other things here. The biology is more complicated in these kind of species, these kind of animals.
Did you ever think that, for instance, long-living colonies could be better objects for such a study? Because you worked with species that are quite short-living.
Yes.
Yeah, but if you imagine, for instance, cyclostomes, Antarctic
yes.
cyclostome that living tens, maybe more years,
yes.
then you could sort of compare different parts of the colonies and see this trajectory.
I think that cyclostomes, there may be more potential for a couple of reasons. One, because they are longer living, but also because they are biologically a little bit more simple. They are not as evolutionarily advanced as these cyclostomes, and they seem to reflect what is going on in the environment more easily in their skeletons. Biology doesn't get in the way so much. So there may still be some potential with this. I was very limited to these short-living species because this area, because of the glaciers, the iceberg scour means that it's almost impossible to find any colonies that are more than 1 year old. I think it's published it's a 96% death rate every year for bryozoans, so all of it is scraped clean and then new ones come in. So it's really hard to find numbers of larger, older colonies. But yes, it would be interesting to do, but difficult.
Also, those long-living stuff normally inhabit like 100-200 meters depth.
Yes.
So they're living much deeper, and it means the temperature is much more stable there.
Exactly. So you would be really lucky to see any variation there at all, I think. So yeah, it's a tricky one. I think bryozoans are not the best choice for a few reasons, and this is one of the reasons for this area of the world anyway. It's probably a reason why people study planktonic animals and things like this, because you really need to do a large amount of replication to see these patterns, to see if there is any variation naturally in these patterns. In the past, all work that has been done on bryozoan skeletons, they have done maybe 2 or 3 replicas, and they think they see a pattern, but it's not enough, because there is so much variation when you start looking at hundreds.
Well, on your underwater images,
Mm-hmm.
there was some sort of whitish meshwork on the water, on the bottom, on the hard water. What was that?
Ah, okay. I think. Let me see if I can find it. This. Oh, hang on. This stuff here. This
That's
kind
some
of
other, yeah,
white
and
fluffy
other
stuff?
images.
and like here.
Yeah, like
this
here.
stringy stuff.
Yeah.
This is worms. This is the tentacles from these really big worms. I can't remember their names.
I mean, polychaetes.
Yeah, polychaetes. Yeah, they're really
Okay,
common.
that's fine. Oh, and then I missed. Why do divers use the second rope for?
So it's very stupid. I don't know. You have one rope with your buddy, so that you always know where they are. If it's very dark or if something happens, you can just pull and you have them. And then you have the rope to the boat,
Okay.
because the people in the boat are always in control. So the divers are
And the
not
second
allowed to
diver
decide.
also has two
He
ropes.
has the same, yes. So 2 divers and
Okay,
2
okay.
more ropes to the top.
So do you have any recorded killer whale attacks there?
No, this has never been seen by anyone, I don't think, any deaths by killer whales. I think it is paranoia more than anything else. Especially at this site, they are very afraid because there was a death from a seal.
Okay.
And we have many rules and regulations about diving for science in the UK. And even though this is Antarctica, we have the same laws and rules. And they said, okay, you had 1 death, this is very bad. Any more accidents at all, no more diving ever.
I've heard the story, I mean, a legend from this area that
Yeah.
leopard seals biting the balloons of Zodiac from behind.
Oh,
Is it true?
I haven't seen this.
People
Yeah.
were telling that they would put buckets on the balloons.
Ah, okay. I would believe it. Yeah, I haven't seen this. Thankfully, but they are leopard seals. I think are maybe a bit misunderstood as well, because we think they are always aggressive, but actually sometimes they are just curious or playful. But yes, they're just afraid because we have this one death.
Well, actually, in Antarctica, I've seen how walruses
Yes.
can kill any
Yeah, well, it's a
balloons
different
or
story.
everything, and I think, well,
Then
I wouldn't.
Yeah, walruses are nasty. Yeah.
But the last, I mean, diving question. The first image you had, is that some kind of weird system of floats?
Oh yes, no, this is very strange. This is
How
just.
does it work?
It's just this. We only use this in the UK.
Yeah, the first
actually.
one. Yeah.
This one here.
Yeah.
So, I don't know if you can see it very well. Yes, it's just because in each of these bags was about 50 kilos of rocks, and I'm not so strong. So I wanted to pick these up and take these with me, and using these balloons, if you like. These are actually surface markers to alert people if you have a problem. But we use them just underwater, just to take some of the weight away. So I could just lift it easily and take it with me.
Were you attached to this construction?
No.
It's very dangerous.
Yes. No. I was not attached to this. No, of course
Okay.
not.
And the last question, kind of
Yes.
more extended.
No problem.
And you didn't say anything about how these creatures calcify, because when you think of magnesium percentage, you need to understand first how the
How they do it.
calcification takes place.
No, it's true. It's a problem we have with bryozoans, because we are working backwards, because we don't know any of this. We don't know any of the proteins involved, any of the genes. This is all completely unknown.
Well, at least about compartments.
Oh yes, we know. Yeah, we know a bit about the compartments. Yes.
What is pH?
Yeah, it's a bit different. Well, we've only looked in a few species, and the work was done back in the 70s, I think, and I don't think anyone's done any new work since then. But yes, it seems to be a bit different in the different species as to why they are changing the magnesium and how they are doing it exactly.
No, I'm asking how the calcification takes place.
Okay.
In bryozoans.
Okay, so they put out a sheet of cells first, and then they have this one large cell, if you like, the calcifying cell that comes and absorbs that, and then it kind of grows around that, if you like. It's, yeah, I could draw you a picture or give you a paper.
I have one more question.
Mm-hmm.
Why the experimental study has not been considered, like putting these bryozoans in different treatment conditions and looking at the magnesium content?
We have done some of this. Well, it's not been done in bryozoans, it's been done in other animals. And this is quite popular. The reason we didn't do it this way, there's two reasons really. The first is bryozoans are very hard to grow in aquariums. They don't always like it. And it can be a bit slow. So yeah, that's kind of the two main reasons why we didn't do it this way. But the other reason as well is because really if you do the aquarium experiments, you need to also do something in the sea to prove that this is accurate, because in the sea it's not only temperature that changes, and it's not as constant, of course, as you have in the aquariums. So it's just a different approach really that we took to doing it. You can do things in aquariums also, but I don't think anyone's had any success trying to do it on bryozoans yet. Yes.
Might isotopes be useful in this search, in this quest?
Yes, some people use isotopic analysis instead of magnesium, and they can also be useful to give another measure of what's happening in the sea at the time when the skeleton was deposited. And yes, it's definitely possible, it can be done. I'm actually looking to, I still have all my samples, and so I'm looking to do some isotopic analysis on the same samples, so that we can maybe compare them and see if we can learn something more.
It would be extremely interesting to look at the variation.
Yes, it would be, you're right. See if the variation
you demonstrated
is similar.
something really wild. I mean, like how it corresponds with isotopes. I think
Yeah,
it's interesting.
it's a bit strange, yeah. Yes. Yeah, it would be interesting to do the isotopes though, I agree.
Okay, I suggest that any other questions, can be asked during the break, and we'll be looking forward to the second part about the invasive species in the UK.